more recent data from earlier this year. We are examining the drug and its effect in pancreatic cancer. And this is results from a phase 2 trial. Here, this is PET scans of a patient who has metastatic pancreatic cancer. So the cancer has spread to the different organs and the lung in particular and the liver. And you see here, after 2 cycles of treatment, that these hot spots in the liver and lung have disappeared. So this is metastatic pancreatic cancer. It's a very difficult disease, as you all know. And Taxol is not used for the treatment of pancreatic cancer because it's not really active. Gemcitabine, on the other hand, is the standard of care for these type of patients. A typical response rate for gemcitabine alone is only 8%, and median survival of these patients is less than 6 months. But when we combine nab-paclitaxel or Abraxane and gemcitabine, your response rate jumps to 46%, and you more than double the median survival to greater than 12 months. So this is, again, telling us that there's something unique about this nano form of paclitaxel that makes its way to the tumor and can show benefit to patients. So this also brings up or opens up the opportunities in the clinical studies to look at personalized medicine. And that should be one of the key focuses of nanotechnology and nanomedicine-based drugs. Because usually you will try and target a drug to a certain location. And so you can interrogate the tumor or the type of tumor for the patient, look at what markers are expressed, and therein you can use nanodiagnostics for that purpose. Once you get that information, then you know how to treat the patient and make a decision on therapy. So these early studies that we have done suggest that we could use personalized medicine in the treatment of these patients. And we interrogate the patient's tumor for the protein spark, and then based on the spark, we can treat the patient. And here, these are animal patients where we first did this. This is in tumor xenografts. There's a low spark expressing and a high spark expressing tumor. Both of these are grown up in the tumor. This is the control. But when we treat them, the blue is the one with high spark and red is the low spark. It's the same tumor. Treated with the same dose of Abraxane, you get a better result with the higher spark tumor. So this was the first suggestion that we could do personalized medicine with Abraxane. We did the same thing in patients. So in the pancreatic cancer study that we looked at, for all of the patients, we got their spark signature. And we could divide up the signature based on their spark level into what we call a low-risk group and a high-risk group. And if you examine the survival of the patients, you can see that this group with what appears to be higher spark in the green showed a much better survival, 13.6 months, compared to the group which has lower spark, which was only 8 months. So this is a good example, a preliminary data, and we're trying to validate this effect in a phase 3 study in pancreatic cancer. What's important to note also that even this group, which had the worst prognosis, had a survival of 8 months, which is better than gemcitabine alone, which is the standard of care. So as far as the clinical results, we've done many different clinical studies. There's probably 80 to 100 clinical studies ongoing with Abraxane in different combinations and different tumor types. But the major studies were, of course, the breast cancer study, which I showed you. We just finished a non-small cell lung cancer study, which was positive, and that was also Abraxane versus Taxol in combination with carboplatin, and we see better results, a better response rate for the Abraxane arm. This data was just released at ASCO earlier this year. And we have strong evidence of activity in the phase 2 setting in pancreatic cancer and metastatic melanoma. So, can this technology be applied to different drugs, or is paclitaxel the only one that can benefit? And so here I show you a pipeline of different drugs that are in development. And these are 5 different drugs. This is docetaxel, which you're all familiar with, is almost completed phase 2 studies. We have an mTOR inhibitor, which is rapamycin, again in the nanoparticle or NAB form, which is close to completion of phase 1 studies. We have a HSP90 or heat shock protein 90 inhibitor that is starting phase 1. We have some other unique compounds. This is a vascular targeting agent that's about to start phase 1. And we have another novel taxane that's about to get into the clinic. So we have 5 other products that we've either discussed with the FDA or have filed INDs on. And there's no real surprises now because the FDA understands the concept of our product with their experience with Abraxane. So the second time, it's much easier, and they understand the constructs, they understand what we're trying to do. So even though we're working on different molecules, the process is smoother the second and third and fourth time over. So with that, I'd like to end, and thank you very much for your attention.